this is how a bacteria looks like. We talked about flagellum. So it is you can call it, it's like a motor that helps the bacteria to move. We talk about fimbria. Fimbria, they are the appendages that on the surface of capsule, they attach to the surface. And we talk about cell membrane. We talk about cell wall, and this is the capsule on the surface. So they look like this. The electron micrograph looks like this. Nuclei. Now that's the reason we call that prokaryotic cell. Prokaryotic cell because they have a primitive nucleus. We call this nuclei because they only have chromosome. They are not surrounded by a membrane. So you only see chromosome bathing in the cytoplasm together. That's it. We call it nuclei. You remember Android? Android phone. What does it mean? Oil. Oil. Oil means like. So this is called nucleus-like structure. Android meaning that it's like human-like structure, human-like being. Nuclear means like nucleus-like structure. We call it nucleus-like because it is not a nucleus per se, because it does not have a membrane surrounding it. The chromosome is like bathing in the cytoplasm. Ribosome, they are for protein synthesis. This is something that you have to memorize. You will see it repeatedly. It is important. Protein synthesis is a very important process. We'll have a lecture dedicated to DNA replication and protein synthesis. And I guarantee that I'll test you on that in the exam in the future. Thylakoid. Thylakoid, they are light-sensitive pigment in the cyanobacteria. You will see thylakoid in the plant, in the chloroplast. They are also called thylakoid. Some bacteria called cyanobacteria or blue-green algae, they can do photosynthesis. The reason they can do photosynthesis is because they have a light-sensitive pigment in the thylakoid. So not all bacteria, they are harmful. They can make their own food, some of them. So we talk about ribosome. Ribosome, they are inside the cytoplasm. They can make protein. We talk about nuclei. They are the chromosome that are lying in the floating in the cytoplasm. And then we talk about what did we talk about previously? Ribosome thylakoid. It's not shown in here, thylakoid. So these are the three characteristics. They have simple structure, metabolically diverse, meaning that they adapt to most type of environment. This metabolically diverse is one of the reasons that contribute to that they can adapt to different type of environment. It is also one of the reasons, metabolically diverse is also one of the reasons that they can generate a resistance to different drugs. They can adapt to different environments. Not only they are metabolically diverse, but because they have three layers of protecting them. I told you the capsule is the one that can resist a lot of harmful substances to them. So this is just a summary table of what we just talked about. Eukaryotic cell that has a true nucleus. So they have a true nucleus because the nucleus has a membrane surround all the chromosomes. They also have membrane-bound organelles. We'll talk about different organelles right now. Eukaryotic cell, they made of animal, plants, fungi, and protists. Protists, they are the like amoeba or paramecium. Some eukaryotic cells have cell wall. For example, plants, they have cell wall. The cell of the plant, they are made by cellulose, like the leaves. For the fungi, they use cellulose and chitin. And Lignin is another substance that makes up of the cell wall. So for fungi, you have chitin, and then chitin is also the one that make the insect exoskeleton. You know, when you crush a cockroach, you know, it sounds crunchy. Why it sounds crunchy? Because they use chitin to make their exoskeleton. That means the outermost is the shell, the hard part. So fungi use cellulose and chitin to make the cell wall. Insect exoskeleton is mostly chitin. For the leaves or most of the tree or plant that we see, they are mostly use cellulose. Lignin, plants use lignin to make the xylem. We will talk more about plant later in the semester. So this is the summary of the eukaryotic cell. We'll go over each one by one right now. Yes, a lot, yes. So organelles. I always tell you that each cell has an organ in it. The correct term should be called organelles. So organelles, they are the small, tiny organs of each cell. They have a different function and structure. They use a factory as an analogy because different departments of the factory, they make different things. Similarly, different organelles, they also perform different functions. Okay, this looks daunting. I may put this picture in the exam and then ask you to label them. But of course, the labeling will be in the form of multiple choice. So in other words, I want you to be able to identify the shape or the structure and the name of it. But the spelling is okay, it will be multiple choice. So at least you need to know, okay, this is the nucleus, inside the nucleus. The purple ball is the nucleus, inside the nucleus is the nucleolus. And then the nucleus is surrounded by the nuclear envelope or nuclear membrane. And then outside of the nucleus, you have the endoplasmic reticulum, specifically we call it rough ER. Rough ER is attached directly to the nuclear envelope. And then you have smooth ER, that is in the cytoplasm. You have ribosome attached to the rough ER. And you also have ribosome freely floating in the cytoplasm. You have mitochondria, looks like this, very, very important. Peroxisome, well, it's just generic structure, looks like this. You have Golgi apparatus, that is close to the cell membrane. You have lysosome. Cytoplasm is the liquid in the cell. You have centrosome and centriole. We will talk about the relationship of centrosome and centriole later. You have regular vesicles, yes. Even animal cells will have vesicles. You have cytoskeleton. Cells, they need to have a skeleton to maintain the shape, and they also use the skeleton to transport material within inside the cell. So you have microtubule, actin filament, and intermediate filament. You have probably 3 types of cytoskeleton. On the cell membrane, I told you that you have a transmembrane protein that can go through the membrane, and also you have phospholipid. We'll talk about all this right now, one by one. This is what a cell looks like under the electron microscope. Plant cell. Again, I can put this picture in the exam and ask you to label cell structure, but again, you will be in a multiple choice. For the plant cell, you have a central vacuole. Central vacuole is something that animal cells do not have. Cell wall. Cell wall is also another structure that animal cells do not have. Last but not least, the chloroplast. Chloroplast is the 3rd structure that animal cells do not have. I forgot to tell you, these 3 are important. Okay, previously I want to tell you that there are also things that plant cells do not have in the animal cell. Let me see which one. Centriole and centrosome. Plant cells do not have central and centrosome. And then peroxisome. Plant cells do not have peroxisome. So there are two things that in the animal cell plant cell do not have, but there are three things in the plant cell that animal cell do not have.